Welcome to the endocrinology simulation. In this simulation you will dive into the function of hormones by controlling the fertility of both male and female rabbits. Are you up for the challenge of developing a hormonal contraceptive that can work on rabbits? Find the lockers and open one of them. Find a lab coat inside the lockers and pick it up. Well done. Now find a glove box and click on it to wear gloves. The zookeepers are discussing the possibility of keeping male and female rabbits in the same pen to save space. Let's go to the zoo.
It's great that you could join us.
Yes, good of you to join us. We're having this issue where it is difficult to control the population of rabbits when males and females are kept in the same pen.
Could we not keep them in different pens?
We've had accidents where little children have set one male loose in the female pen. You can imagine what happened.
What about giving them a hormonal contraceptive?
That might work. Could you please help to design the best contraceptive for our rabbits?
Could you help the two zookeepers develop an efficient and safe hormonal contraceptive for the rabbits in the lab?
That would be amazing. Go to the feeding room to learn about the endocrine system and start the development of the contraceptives.
We'll make sure that we have all the relevant certificates and approvals needed before performing these experiments.
Click the door at the end of the petting zoo to go to the lab room. Welcome back to the lab. Before starting with the hands-on work, let me show you some of the more important hormones responsible for controlling fertility in females. Go to workbench 1. On the PC screen you will see a full diagram of the hormonal pathways. However, I'll unravel the diagram step by step with the conversations, so you can follow the process. You can also see additional information on the hollow table to your right. Remember to check both of them to better understand the explanations. Let's start with the pituitary gland. It sits next to the hypothalamus. A stimulus will cause the hypothalamus to release gonadotropin hormone. The hypothalamus and pituitary glands are connected by a blood vessel. The GnRH will cause the release of follicle-stimulating hormone and luteinizing hormone in the anterior pituitary. The hormones are then cycled in the body via blood vessels and the heart. The increase in the previously mentioned hormones lead to the release of estrogen and progesterone in the ovary. High concentration of estrogen and progesterone may trigger a positive or a negative feedback in the hypothalamus and pituitary. If high concentrations of estrogen and progesterone occur just before ovulation, it will trigger a positive feedback in both hypothalamus and the pituitary glands, increasing the concentration of LH, estrogen and progesterone. The sudden increase in LH triggers the ovulation in humans due to a complex series of reactions. However, in any point of the menstrual cycle different than just before ovulation, high levels of estrogen and progesterone will trigger a negative feedback. Negative feedback loops are important factors for controlling hormonal levels. Increased levels of a steroid hormone, such as estrogen or progesterone, will signal to the hypothalamus and the anterior pituitary to slow down their production of GnRH or LH and FSH respectively, which consequently will result in a decrease of the levels of estrogen and progesterone. If hormone A causes the production of hormone B, which of the following is an example of a negative feedback mechanism? Well done, you spotted the example of negative feedback. As you clearly know, more of the controlling hormone will decrease the production of the controlled hormone during negative feedback. Which of the following statements is true? That's right. By negative feedback the steroid sex hormones suppress secretion of those three hormones. Which of the following would decrease FSH secretion? Sure, FSH production is reduced when there's a high amount of androgens in the blood. Amazing. Now I'm sure you know all you need to start the development. Now click that big double door over there to go to the feeding room downstairs. Click the highlighted door to go to the feeding room. Alright, now we've had a look at the basics, let's try to put all this theory to good use, and see if we can control the fertility of these rabbits with hormone treatments. The rabbits in front of you are the ones you will test the treatments on. Today you will use 3 different treatments, one with progesterone, one with testosterone and one with gonadotropin-releasing hormone, or GnRH, for short. The treatments will be added orally to the rabbits, but you don't need to worry about preparing them. I have already mixed them with banana and placed them in the syringes on the workbench. You can point at them to see what's in each syringe. We always need to have control treatments. We need to ensure that the control treatments contain none of the active ingredients and take into account methods such as how the treatment is administered. To supply testosterone and progesterone, we need to use sesame oil to dissolve them. However, gonadotropin needs to be dissolved in physiological saline. How can we control for the possible effects of the stress of oral injection and the injected oil when evaluating the effects of treatments 1 and 2? Exactly. We want to make sure the effect does not come from the way the treatment was administered to the rabbit. How can we control for the possible effects of the stress of oral injection and the injected saline when evaluating the effects of treatment 3? That's right. When creating the control, we want to ensure the rabbit reacts differently to the hormonal treatment only, and not to the way the treatment is administered. These rabbits were forced through puberty over the last week by using estrogen injections. This is to ensure they have the same reproductive history with no former pregnancies. On the workbench you will find a syringe with a treatment for each of the rabbits. When you have administered their first treatment, I will return them to their cages and take care of the rest of the rabbits. I will also take care of the repeated treatments, as my calculations show the treatments need daily repeating for 10 days to show any measurable effect. Make sure to feed each rabbit with one treatment. I'll keep track of which rabbit is fed what and take care of the control treatments. First, feed one rabbit with treatment 1. Treatment 1 contains progesterone dissolved in oil, mixed with banana. Feed treatment 1 to a rabbit. The next syringe is treatment 2. It contains testosterone dissolved in sesame oil and mixed with banana. Wow, these rabbits seem to really like banana. I wonder if Wilco and Anna, the zookeepers, also like it. Feed treatment 2 to a rabbit. Now feed the last one with treatment 3. That's delicious banana mixed with gonadotropin-releasing hormones dissolved in physiological saline. Feed treatment 3 to a rabbit. That's 3 stuffed rabbits right there. Well done. We will need to take blood samples from the rabbits to measure the hormone levels in the blood. If you could take the first samples to show me how it's done, then I can make sure to sample the blood when I do the next treatments. I will store, save, and label all the blood samples. You made that look super easy. Now I'm sure I can do it, so, if you don't mind, I'll do the rest for you. It's time to go to the microscope room where we can have a look at the effect of the treatments on the uterus. I'll meet you there. Click the door to go to the microscope room and fast forward time 10 days. After you injected the first set of treatments, I continued them for the following 10 days. Yes, 10 whole days have passed. A way to test for fertility in female rabbits is to examine the morphology of the uterus. If the rabbit is either ovulating or pregnant, the endometrium in the uterus will be enlarged and thickened. As you remember, whenever the rabbits got injected with a treatment, we extracted blood. I sampled blood from the rabbits every 2 hours for 10 hours after the first injection. We will use that blood to check the hormones levels using a radio-immunoassay later. But now let's focus on examining the morphology of the uteri from the rabbits. In the meantime, I will make sure that the blood samples are ready to be analyzed. I have just prepared some samples of rabbit uteri from different treatments in the back room for you. Have a look at the image to see the uterus placement in the rabbit. For now, let's have a look at the uteri under the microscope. I have prepared some microscope slides with uteri from different stages during a cycle. On the screen on the workbench you can see the McPhail scale represented with images. This scale is used to score the thickness of the endometrium in the uteri. A high number, such as 5 or 6, indicates a thickened endometrium, which is necessary for the fertilized egg to settle in the uterus. A low number, 0 or 1, indicates a thin endometrial layer. Oh no, I forgot to label the slides and now I don't remember which one corresponds to each development stage. Why don't you have a look at them and score the uteri from the rabbits? Luckily, we have a reference of the scale. You can compare what is on each slide to the reference images shown at the bottom of the screen. Just click on one of the 6 small images on the screen to indicate the correct state you are observing. Grade each slide using the McPhail scale. You're right, that looks like a 5. Sure, let's mark that as a 1. Click on the lab pad for more information. Yup, that looks like a 3. Yes, I would also have scored that one as a 4. Yeah, I would say that's a 2. I agree, that looks like a 6. Nice. You managed to score some of the uteri using the McPhail scale. I have already scored the rest and summarized the uteri scores in a bar chart. Have a look at the screen, the bar shows the average and the blue line shows the interval minimum to maximum. I really want to understand that data. What is a reasonable conclusion you can draw from it? You can have a closer look at the graph on the screen. Yes, perhaps due to their ability to suppress LH and FSH levels by negative feedback. You can also define the hormonal status of the uterus using the blood from the rabbits we collected before. To have a go at this, go to the radio immunoassay room, down the hallway and to the left, where you will perform a radio immunoassay. Now, go to workbench 2. Radio immunoassays rely on the use of radioactively labeled antigens. I know it sounds difficult, but it's a very simple technique. To ensure no radioactive contaminants get spread, we keep a portable contamination monitor on the workbench. You don't need to do anything with it. If we spit any radioactive substance it will beep, but I am sure that you can pipette properly. To perform a radio immunoassay we only need 3 things: the antigen we want to detect, which in this case is the hormone we want to know the concentration of, antibodies that bind to the desired antigen with high affinity, and radiolabeled antigens that will be used to compete with the antigen in the samples. This will be used to determine the final concentration of the antigen of unknown concentration. Now for the fun part. In order to calculate antigen concentrations accurately, we need to prepare a standard curve with known concentration of unlabeled antigens before examining the sample. First we prepare a mixture of radioactive antigens, often we use some form of radioactive iodine. Prepare a tube by adding the anti-LH antibody with the pipette. Add antibodies to the empty tube. Okay, that first step was super simple. Now we add a known amount of antibodies without any radioactive labeling. Just take a fresh tip and add the radio-labeled LH. Add the radiolabeled LH. Then we do the same procedure with increasing known concentrations of the antigens to create a standard curve. I already did these steps. Then we do all the steps again. This time we will do it with our original blood serum, instead of the unlabeled antigens. Add blood to the green tube. Simple, wasn't it? Now all you need to do is measure the radioactivity in the Geiger counter on the workbench. Measure the radioactivity. Inspect the graphs available on the machine screen. I asked you to make a prediction about fertility, and earlier you chose only high progesterone because this will inhibit LH and FSH secretion. High progesterone levels did indeed inhibit LH and FSH secretion. Did you notice testosterone did so too? Let's have a look at how the treatments affected average serum levels of LH and FSH over time. Check out the graphs. Toggle between the graphs. When you are ready, I have a question for you about them, so make sure you pay attention. If you need to, you can lower the lab pad to have a second look. Based on the graphs, a reasonable conclusion would be: you are awesome, well spotted that the testosterone and progesterone affect LH and FSH by negative feedback. Go to Workbench 1 to learn about fertility in males. Let's learn about the hormones in the male body. As in the females, GnRH is produced in the hypothalamus. Check out the image on the screen and click Continue in the lab pad to continue. Similarly, FSH and LH are produced as a result of a signal coming via the bloodstream from the hypothalamus to the pituitary gland. Check the new information on the screen in the hollow table. Luteinizing hormone stimulates testosterone production in the Leydig cells. Additionally, the follicle-stimulating hormone stimulates the production of the inhibin hormone in the Sertoli cells. In the testes, increased levels of testosterone and FSH acting on the Sertoli cells stimulates spermatogenesis. On top of that, as shown on the screen, inhibin secreted by Sertoli cells exerts negative feedback on the FSH and LH secretion in the pituitary gland, consequently causing decreased spermatogenesis. Phew, that was a lot of information. Let's go to the feeding room to prepare our experiment. Click the double door to go to the feeding room. Now let's use the concepts we have learned. We will use a compound called dimethandrolone undecanoate, or just DMAU for short. DMAU is an androgen steroid with antigonadotropic effects. This means that it can suppress the production of sperm. The male rabbits are fed the treatments via tablets mixed in with banana for 12 weeks. We will use a concentration of 5.0 milligrams per kilogram and day. Feed one of the two males with the DMAU treatment. I will take care of the control. Steroids and androgens play the largest role when controlling male fertility. Testosterone, progesterone and estrogen are in this category. Of the 4 compounds below, 3 will suppress male fertility, and 1 will not. Which one will be unlikely to suppress male fertility? Just that, FSH will, in fact, increase the fertility of males. Let's test if DMAU works as intended. I will take care of feeding the rabbits the rest of the experimental weeks, and I will also collect blood and semen samples every 2 weeks. You can look at the results back in the lab. Click the door to go to the microscope room and fast forward time 12 weeks. Amazing! We are back in the microscope room. We will look at the fertility of the male rabbits. As you can see in your lab pad, 12 weeks have passed, and rabbits have received DMAU doses regularly. Let's use the microscope to analyze the effects of DMAU on the rabbit's semen. Transfer a drop of rabbit semen from the Eppendorf tube to the Neubauer chamber. The Neubauer chamber is very similar to a standard microscope slide, except it has wells in it. That allows us to precisely count the number of semen cells per milliliter. Let's have a look under the microscope. Place the Neubauer chamber in the microscope. We need to count the number of sperm cells in each of the small squares in the middle of the image. It is a bit difficult to see, isn't it? Click on the microscope view of the Neubauer. This is better. Here you can see a 3D representation of the sperm cells that we have in the Neubauer chamber. I just zoomed into one of the squares that you see on the screen. Take a look at the hollow table to see it. I just finished counting. I know, I am fast. Look at the graph on the screen and click on it to see the rest of the data. That was it regarding sperm count. Let's analyze the concentrations of the hormones. According to the data shown in the graphs, which of the next statements is true? Great, you are amazing. That's definitely a reasonable explanation of the graphs. So now we know that DMAU can be used as a potential birth control method for males. Testosterone injections have almost the same effect as DMAU. However, high doses of testosterone have unwanted side effects, such as weight gain and hair loss, which are not seen with DMAU. Some studies have even shown that using DMAU can potentially reduce the loss of bone mass. Amazing. We should share our findings with the zookeepers. Click the door to go back to the petting zoo.
Great work. I need to prepare another fertility experiment for the next user. Could you help me by answering a quick question? Based on your results, what should we investigate next to obtain the best fertility inhibitor for rabbits? WOA. You are a clever one. That's definitely a good suggestion for a follow-up experiment. I'd be excited to see the results from that one.
The hormones we tested today are often present in both old and cutting-edge hormonal contraceptives. Some of the early experiments used progesterone and estrogen extracted from animals. DMAU is still in an experimental phase and is being tested for extended usage. Hormonal birth control is a relatively new invention compared to other forms of contraceptives. The combined pill, or just the pill, has been widely used from circa 1960s as a hormonal contraceptive for women. Tests began with steroid hormones as early as the 1930s, and have continuously evolved through time into multitude of versions that are used by many millions of women around the globe today. To this day, no viable male alternative has gained wide acceptance. DMAU is a promising candidate, but it is still not perfected, and thus, not approved for use. Progesterone and testosterone both function to inhibit the fertility of the female rabbits. Additionally, DMAU worked perfectly to inhibit the fertility of male rabbits.
Wow, you surely learned a lot. Thanks so much for passing it on.
Yes, thanks. Now we can keep these rabbit numbers in check.
Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.